Welcome to Curiosity, the podcast where science meets wonder. Join us as we explore the fascinating, the innovative, and the unexpected, from the secrets of the cosmos to the marvels of everyday life. Each episode takes you on a journey to uncover the knowledge and stories that fuel our endless quest for understanding. Let your curiosity guide you.
Every single day, your brain is performing what really feels like an unbelievable magic trick.
It really is.
It takes all these passing impressions of your life. You know, the conversation you just had, an article you skimmed, the frustration of being stuck in traffic, and somehow it selectively converts just a tiny fraction of those moments into what we call memory.
And these memories, they're the fundamental building blocks of who you are. They're the data that guides, literally every decision you make.
It's astonishing when you stop and think about it, just the sheer relentless volume of sensory data that's bombarding us every waking second.
Oh, absolutely. If the brain actually stored everything, we would be, instantly paralyzed by information overload.
Right.
So the brain has to be an absolute master of triage. It must have some kind of sophisticated system for deciding, okay, what do we keep and what do we just let go?
But the frustrating reality, and I think anyone who has tried to, learn a new language or master a new skill or even just cram for an exam knows this, is that most of those experiences they fade.
They dissolve back into the noise.
Exactly. So the core question for neuroscientists, really for decades, has always been, how does the brain decide what information is worth keeping? And maybe even more crucially, for how long?
Yeah, why does that complex concept you spent 3 hours studying last week just vanish? But, that vivid, highly emotional memory of some brief event from a decade ago is still crystal clear.
That's the mystery.
That core mystery, the science of memory durability, is what we are really going to explore today. And it's all fueled by some genuinely groundbreaking research.
And we have one foundational source for this conversation today. It's a detailed summary of some landmark work from Rockefeller University, and it was published in the journal Nature. And it fundamentally changes how we understand memory persistence.
It really does.
And the big reveal here is that the lasting quality of memory is absolutely not governed by some simple instantaneous switch. So if you're still thinking about long-term storage as like flipping a light switch on, you need to forget that analogy right now.
Precisely. What these researchers uncovered is not a single decision point. It's a finely tuned, layered series of molecular programs.
A layered series.
Yeah. And these programs, they act like sophisticated sequential timers that determine how durable a memory is over these really extended periods. It's a stunningly coordinated biological dance of molecules activating in different parts of the brain at different times to build permanence step by molecular step.
So our mission in this exploration is to give you a clear, thorough shortcut to understanding this incredibly complex stepwise system.
Right.
We need to look at what the old memory models got wrong, and then we'll zero in on the key brain structure that acts as the sort of decision-making and routing hub. And it's probably not where you think.
And then we'll get into the specifics. We'll introduce you to the actual molecular timers, the genetic regulators themselves that control whether your memory lasts for a week, a month, or a lifetime.
It's an exploration into the molecular clockwork that literally defines our capacity for learning and for building our own personal history.
It's fascinating stuff.
Okay, so let's unpack this and maybe start with a bit of the scientific history. Before this recent work came out, when neuroscientists talked about how memories were stored, what was the established wisdom? What was the accepted framework?
For decades, really, the field relied very heavily on what we call the classic two-center model of memory storage.
Two-center model.
Exactly. It offered a very neat, very clean geographical picture of the whole process. On one side, you had the hippocampus.
Right. Everyone's heard of the hippocampus.
It's famous for that curved seahorse shape deep inside the medial temporal lobe. And it was understood to be the primary staging area. Its job was to support the initial formation and retention of short-term or temporary memories.
So think of it as like the brain's high-speed temporary buffer.
That's a great way to put it. Yeah, holding information for minutes or maybe hours.
Okay. So it's where the data gets processed immediately after intake. And then according to this old model, what determined if that temporary data became, permanent knowledge?
Well, the idea was if the memory was somehow flagged as important, maybe because of repetition or it was emotionally charged, it was believed to be transferred or shuttled through a process we call consolidation.
Right, consolidation.
And it would move out of that fragile hippocampus and into the cortex. that vast, wrinkled outer layer of the brain, which was seen as the permanent storage facility for long-term memories.
So the cortex was the hard drive.
The hard drive, exactly. The idea was that the cortex provided this robust, physically dispersed network that was necessary for decades-long recall.
And within that model, the mechanism for achieving that permanence was also pretty simple, which I guess is why it stuck around for so long. It led directly to what the researchers call the on-off switch analogy.
Precisely. I mean, the older prevailing view held that long-term memories sat behind these simple biological on and off switches at the synaptic level.
So once the memory makes the journey to the cortex, it's consolidated, and boom, a switch flips.
That was the idea. A single, decisive molecular event flipped the switch from temporary to long-term, and that was it. The consolidation process was over, the memory was stable.
And the researchers even had a specific term for this, right?
They did. They mentioned transistor-like memory molecules that supposedly acted as these instantaneous on-off switches. It offered a binary choice: stored forever or discarded now.
You know, that model is beautifully simple. It's very intuitive. But if it was so useful, why did it need to be revised? What was the essential flaw, the explanatory gap that this new research is designed to fill?
The limitation was fundamentally the durability spectrum.
The durability spectrum.
The classic model, while it was really valuable for explaining memory location, it failed completely to account for the enormous demonstrable variance in memory longevity that we all experience every day. I mean, if memory storage were truly a one-time flipping of a switch, every single memory marked long-term should, in theory, persist indefinitely and be equally robust.
But that's just not true in our own lives, right? When we think about learning and recall.
It is entirely disconnected from reality. Think about common memory phenomena that simple switch model just can't explain. For instance, why is it that if you cram a lot of complex neutral facts, say historical dates, into your brain in one night?
Ah, the all-nighter.
The classic all-nighter. Why do those memories often vanish completely within, say, 72 hours? Even though they technically were long-term enough for you to pass the test the next day.
Right. They were definitely consolidated. They definitely left the hippocampus, but they didn't stick.
Exactly. And you can contrast that with something like the distributed practice effect, where reviewing information across several days leads to far superior long-term retention.
The old model couldn't really explain why that spacing made such a difference.
No, other than just vaguely saying it strengthened the connection. More deeply, it failed to explain why some memories tied to these low-key but frequently repeated contexts like, knowing the location of a specific street sign you see every single day become permanent, while other complex novel experiences fade away.
So the brain isn't just deciding store or discard, it must be deciding store for 3 days, store for 3 months, or store for 30 years.
Precisely. It's not a simple electrical switch. It's more like a highly sophisticated chemical timer. Or maybe a dimmer switch, where the intensity of the light, the memory's vividness, and its resistance to decay is constantly being evaluated and adjusted over time.
And that is the core revelation of this new paradigm shift, which was championed by Dr. Priya Rajastupati, the lead investigator on this.
It is. She posits that memory durability is a continuously evolving process rather than a one-time flipping of a switch. The brain is constantly running these maintenance checks, adjusting the stability and the reinforcement level over time.
So it's basically deciding if it needs to invest more energy into stabilizing the information or
just allow it to decay naturally, which is the brain's default, energy-saving position.
This implies that the physical architecture supporting the memory has to be actively built and reinforced over an extended period. It's not just about forming the memory. It's about sustaining the physical and molecular structures that make it durable.
Absolutely. And that sustained, time-dependent reinforcement happens through a sequence of precisely timed molecular mechanisms that activate in a specific order, rather than a single decisive event. The longevity of the memory is proportional to how many of those sequential timers it successfully engages.
Okay, so if memory stabilization is this evolving time-based process, and it's not just a direct hop from the hippocampus to the cortex,
then we have to identify the crucial geographical middleman.
the traffic controller.
Exactly. We need to locate the decision-making hub that manages this whole sequential stabilization process, which the team initially described back in 2023.
So where does the memory go after its initial encoding in the hippocampus? Where is this traffic controller located? Because if it's not a straight shot to the cortex, there has to be some kind of intermediate staging ground.
And the crucial discovery that really elevates this research pinpoints the thalamus.
The thalamus. Wait, for anyone who's taken like an intro neuroscience class, that sounds like massive scientific revisionism.
It is.
Traditionally, the thalamus was just seen as a simple relay station, right? The sensory switchboard of the brain. It gathers incoming sensory info, except for smell, and just shunts it to the right cortical regions. It's passive, a phone operator, not a decision maker.
That was the textbook definition for a very long time.
So why the massive upgrade in status? What evidence allows researchers to suddenly assign this structure, which is central but, historically just seen as connective, this high level of decision-making authority in memory?
That's where the elegance of their connectivity tracing and their signaling studies comes in. The researchers found really compelling evidence that a specific pathway, the thalamocortical circuit, is absolutely essential for long-term memory maintenance, not just for sensory input.
Wow.
This research dramatically elevates the thalamus's role. It's identified not as a passive relay, but as a central element in this stabilization pathway that connects the fragile, short-term hippocampus system with the robust, long-term cortical system.
So it's actively gating the information flow. It's deciding which memories get to proceed and which ones are terminated?
Exactly. The source highlights the thalamus as the crucial gatekeeper. It performs this vital function of helping determine which memories should be kept and directs them to the cortex for long-term stabilization.
So it's less like a passive switchboard and more like an air traffic control tower.
That's a perfect analogy. It sits between the two main memory airports, the high-traffic, temporary hippocampus, and the permanent destination, the cortex. It's deciding the priority level, the duration of the memory, and the specific routing pathway to achieve that desired level of persistence.
That fundamentally changes our map of the brain. This isn't just about where memories are stored, it's about how they are filtered and routed. This makes the thalamus an active decision maker in the process of memory consolidation. It's determining the fate of the memory.
And the initial research defining this pathway showed that electrical activity, specifically these things called theta oscillations, were crucial in this circuit for synchronizing the hippocampus and the cortex. But this subsequent landmark research went deeper. It asked, what are the molecular processes happening inside the thalamus itself that execute this decision?
So we're moving from the map of the brain, where the decision is made, to the internal biological engine, how the decision is executed chemically and genetically.
It's the difference between identifying that a bridge exists to cross a river and understanding the tensile strength of the materials used in that bridge's construction and the engineering needed to ensure it lasts for a century.
Yeah.
They needed to find the engine that provides the molecular momentum for long-term persistence. And they found that the thalamus is where the first critical molecular timers are running.
So the immediate next question becomes, what are the specific molecular instructions being executed within the thalamus and then sequentially within the cortex that translate an electrical signal of this is important into a decades-long physical structure?
Yeah, the research really zeroed in on the idea that the persistence of a memory is dependent on its ability to trigger a series of these molecular events that stabilize the synaptic connections, essentially locking them in place against decay.
And those molecular events, they discovered, are governed by an ordered sequence of genetic regulators.
That's right.
This is where we get into the truly fascinating part of the science. To study a process as complex as memory stabilization, which, unfolds over hours, days, and weeks, you must need some pretty sophisticated tools. How did they actually conduct this experiment and observe these sequential memory timers in action inside a living, functioning organism?
The methodology was, it was a masterpiece of control and precision. They started by creating a controlled environment for memory formation, using a virtual reality behavioral system in mice.
Virtual reality for mice?
Yeah. And this wasn't just observing mice in a maze. It was a highly controlled sensory environment.
So what did that setup allow them to control that, say, a traditional maze wouldn't?
It let them track brain activity while the mice formed these highly specific, purely contextual memories, often relying just on visual cues and context. So it isolated the memory to specific neural circuits.
So it reduced the noise that smells or complex physical interactions might introduce.
Exactly. And crucially, they could manipulate one critical variable, repetition.
So repetition became their stand-in, their proxy for importance.
That's right. They compared context the mice encountered only occasionally, Low repetition, low perceived importance, with those they frequently repeated. High repetition, high perceived importance.
So, for example, a mouse that saw the virtual blue room 50 times versus a mouse that only saw the red room twice.
Precisely. And this created a perfect, controlled contrast between memories that were destined to be durable and those destined to rapidly fade. And then they could track the molecular changes in the brain regions of the durable memories versus the fading ones.
Okay, so they established a correlation. High repetition correlated with specific molecular activity in the thalamus and the cortex. But correlation isn't causation.
No, it's not.
They had to prove that those molecules were actually required to make the memory stick.
That's the critical juncture. And to prove causation, they needed surgical precision at the molecular level. This is where the really cutting-edge technology came into play. The co-lead on the study, Celine Chen, developed a refined, CRISPR-based screening platform to alter gene activity specifically in the regions they were investigating, the thalamus and the cortex.
For anyone who isn't familiar, CRISPR is often described as like molecular scissors. It lets scientists snip and edit DNA with unprecedented accuracy. Why was that tool so necessary here?
It was absolutely essential because they needed to inactivate or disrupt the activity of specific genes only in the neurons of the thalamus and cortex, and only after the initial memory had already formed.
I see.
They needed to prove that if you take out a specific molecule at a specific time in the sequence, the memory stabilization fails. If they had just used a drug to block a pathway, it would have affected the entire brain. CRISPR allowed them to be hyper-localized, to remove one piece of the clockwork without dismantling the whole mechanism.
And the results were pretty definitive, I take it.
Stunningly so, absolutely. By removing certain molecular programs, they were able to selectively abolish the persistence of the memory, even if the mouse had experienced that event 50 times.
Wow.
This definitively proved that these molecules were not merely present during memory stabilization, they were functionally required. The major finding was crystal clear. Long-term memory relies not on one simple on-off switch, but on a sequence of gene-regulating programs that unfold like molecular timers across the brain.
And this sequential nature is the core concept we need to really unpack here. Memory durability is built in layers over time, with different molecular programs kicking in at different stages.
Exactly. You can think of it as a series of quality control gates that a memory has to pass to achieve true permanence. The early timers activate quickly, within hours of the experience, but they're transient. If the memory isn't reinforced by the next stage, those early timers fade fast and the memory disappears.
Which is the default forgetting process.
That's the default. Conversely, the later timers turn on much more gradually. They require sustained input, like repetition or high emotional salience, to engage. But once they're engaged, they provide the robust structural support, the chemical scaffolding, for those important experiences to persist for years.
So let's focus on the stars of the show here. The research identified 3 specific transcriptional regulators that are essential for preserving memories. Before we dive into them, maybe let's just clarify that term. What exactly is a transcriptional regulator?
That's a great question, because these are the architects of memory stability. A transcriptional regulator is essentially a master switch for the neuron's genome.
A master switch.
It's a protein that binds to DNA and controls the rate at which genetic information, a specific gene, is copied into RNA, which then leads to the production of another protein. So instead of controlling just one single process, a transcriptional regulator controls the activity of many other genes simultaneously.
Okay, so regulating them allows the neuron to execute these complex systemic programs like building a lasting memory structure all at once.
Precisely.
So if they are master switches, turning them on means the cell is executing a highly complex multi-step construction plan. And it's critical to remember these molecules are not required for the initial memory formation in the hippocampus.
Correct. That's a key distinction.
They are only crucial for maintaining the memory's lifespan afterward.
They're the maintenance and reinforcement crew, not the initial builders. And the first 2 regulators they found reside in that decision-making hub, the thalamus, which really confirms its role as the critical early gatekeeper of durability.
Okay, so the first one in this sequence is Camta1.
Camta1 is described as the early timer. It is the initial molecular response in the thalamus to basically say, attention, this just happened. Let's keep the connections functional for a little bit.
So its role is to keep that early memory intact right after it's formed.
Right. Its downstream targets, all the other genes it regulates, activate very quickly, ensuring the nascent memory has a temporary molecular shield that prevents it from simply vanishing right away.
It sounds like a kind of molecular triage, a rapid response mechanism to hold the memory state until the brain decides if the information is worthy of a larger investment.
That's the perfect analogy. Camta1 keeps the lights on immediately after the memory event. But if the input wasn't strong enough, if the repetition didn't happen, Camta1's effects wear off quickly and the memory dies naturally.
But if the input persists...
If it persists, Camta1 is followed closely by the second, more sustained regulator.
And that would be TCF4, the intermediate timer.
TCF4 is fascinating. It's described as the intermediate timer because it doesn't activate instantly like Camta1. TCF4 activates gradually, which suggests it needs more time or more sustained repetition to fully engage its molecular program.
And what's its critical role?
Its role is fundamentally structural. It strengthens cell adhesion and structural support over time, specifically within the connections between the thalamus and the cortex.
Cell adhesion and structural support. So this is where we move from a chemical signal to an actual physical change, stabilizing the wiring of the circuit. Can you expand on what strengthening cell adhesion means in the context of a memory?
Yeah, it's about building a robust physical foundation for the synaptic connections. Memories are stored in synapses, those microscopic gaps where neurons communicate. To maintain a memory, these connections have to be physically resilient.
Okay.
So TCF4 manages the expression of genes that produce proteins responsible for anchoring the synapse in place. It makes the connection between the thalamic neuron and the cortical neuron physically stronger, thicker, and more resistant to degradation.
So it's like moving from a temporary glue to actually laying concrete foundations for the bridge.
That's it exactly. So if Camta1 is the early alarm system, TCF4 is the long-term construction crew that physically reinforces the bridge between the short-term memory pathway and the long-term destination in the cortex.
And the researchers proved how crucial this molecular duo is, right? Oh, absolutely. They specifically noted that when they disrupted both Camta1 and TCF4, the molecular bridge just collapsed. The neurons in the thalamus and cortex failed to maintain their strong, sustained connections, which directly resulted in memory loss, even for highly repeated experiences.
So without those two molecular programs running smoothly and sequentially, the memory never makes it into the durable long-term storage system.
Never.
Okay, so we have the initial timer and the intermediate structural timer in the thalamus acting as the filtering mechanism. But where's the final, ultimate stabilizing timer located? The one that creates that decades-long persistence.
That final, irrevocable step occurs in the destination region, the cortex. The third essential regulator, ASH1L, is located in the anterior cingulate cortex, or ACC.
And that's a region we know is involved in integrating emotion, decision-making, and long-term storage.
Exactly. And this is described as the late stabilizing timer.
And this is where the molecular process gets its deepest claws into the permanence of the memory, without a doubt. ASH1L's job is the most profound in terms of true permanence. It promotes these chromatin remodeling programs that provide the strongest possible multi-decadal reinforcement of memory stability. If TCF4 builds the physical bridge, ASH1L fundamentally changes the architecture of the cells on the other side to guarantee the memory lasts.
Chromatin remodeling, that phrase is technical, but it sounds absolutely critical to understanding memory permanence. We need to go into depth here. It's more than just changing how the DNA is packaged, which is the simple explanation we often hear. What does ASH1L actually achieve by modifying chromatin?
It's about changing the physical access points to the instruction manual of the neuron. DNA isn't just floating freely inside the cell nucleus. It's wrapped tightly around these specialized proteins called histones, forming a complex called chromatin.
So you can think of chromatin as like a carefully packaged spool of thread.
A perfect way to visualize it.
Yeah.
And if that thread is tightly spooled, the genes, the instructions for making memory-specific proteins, are physically inaccessible to the cell's machinery. They are turned off.
And what does ASH1L do to that spool?
ASH1L is part of a family of molecules that chemically modifies those histones. It loosens the spooling structure. It physically remodels the chromatin. And by doing this, it opens up the regions of DNA that contain the genes necessary for maintaining synaptic strength and structural support.
So it ensures that the specific genes related to that permanent memory are permanently accessible for transcription.
Yes. They are locked into the on position, making the memory highly resistant to degradation, regardless of whether that initial electrical signal has long since faded.
That is the ultimate molecular lock. Camta1 keeps the memory alive for a moment, TCF4 physically reinforces the connection, and then ASH1L makes a permanent architectural change to the instruction manual of the neuron itself. This explains the whole spectrum of persistence, doesn't it?
It explains everything. The whole sequence, Camta1, then TCF4, leading to ASH1L, provides a stepwise molecular explanation for why some memories endure for 30 years while others fade so quickly. The brain is investing progressively higher molecular resources based on the perceived value of the memory, which is proxied by repetition or intensity.
And this really underscores that critical implication cited by the researchers. Unless you promote memories onto these timers, we believe you're primed to forget it quickly. So forgetting, in this model, it's not a failure. It is the highly active, energy-saving default state of the brain.
Absolutely. Retention, especially long-term retention, is expensive. It's metabolically expensive. It requires the sustained effort of activating Camta1, then TCF4, and finally the massive genetic investment of ASH1L-mediated chromatin remodeling.
So if the experience isn't deemed important enough, the molecular machinery just doesn't engage the later timers.
And the default decay process takes over almost immediately. It's an incredibly efficient system that ensures only the most relevant, frequently encountered, or highly salient information receives that expensive long-term molecular support that ASH1L provides.
Which means that for us, the intensity or the repetition of an initial experience dictates the starting signal for these 3 sequential timers. It's a molecular explanation for why distributed practice works so much better than cramming.
Right.
Spreading out the input over time gives the brain's internal timers, the TCF4 and ASH1L programs, the necessary time and signal strength to fully engage and lock the memory down architecturally. Cramming just activates that early transient Camta1 phase.
Exactly. You never get to the concrete and steel phase.
If we connect this new understanding of the sequential molecular programs, Camta1, TCF4, and ASH1L, to the broader context of biology, what is truly fascinating is this realization that the brain may not have invented entirely new molecular tools just for cognitive memory.
It seems to be repurposing ancient, time-tested molecular mechanisms that are already used widely across biology.
Repurposing. That suggests incredible evolutionary efficiency. What are these tools used for elsewhere in the body?
This is a really profound insight. ASH1L, our late stabilizing timer, belongs to a large essential protein family called histone methyltransferases.
Okay.
And these molecules are central to fundamental mechanisms that create and maintain what scientists call cellular memory in non-neuronal systems. They are the mechanisms of durable identity throughout the entire organism.
Cellular memory outside of traditional cognitive function. Can you give us the examples the source materials pointed to?
Sure. The source highlights 2 major areas. First is the immune system. The immune system has to perform a kind of memory function. When you get a vaccination or you fight off a virus, the body has to remember that specific pathogen to mount a faster, more effective response the next time.
Right.
These histone methyltransferases are key players in helping immune cells maintain a long-term molecular profile, a memory related to that past threat. The cell needs to sustain a specific genetic identity related to that past event.
That makes perfect sense. The immune system is essentially running a long-term molecular database of past threats, so it needs a mechanism for permanence.
And second, during cellular development and differentiation. When a stem cell is first deciding what it will become, will it be a neuron, a skin cell, a muscle cell? It has to maintain that identity for the rest of the organism's life, which is often decades.
I see.
These histone methyltransferases help cells remember that they become a neuron or a muscle and sustain that long-term cellular identity by permanently setting the appropriate gene expression patterns.
Wow. So the molecular machinery that ensures a skin cell doesn't accidentally turn into a heart cell is the same fundamental type of biological mechanism that ensures you don't forget your spouse's name or where you grew up.
Essentially, yes. The brain is leveraging the most robust, time-tested biological mechanism available for creating durable persistence. Architectural changes to the genome itself.
It's an evolutionary shortcut.
It is. The brain didn't invent a new system for cognitive memory. It simply borrowed the oldest, most reliable, permanent system from biology to write our personal history.
That is a staggering simplification of the complexity of life, and it's really beautiful.
Now, moving from broad biology to human health and intervention, what is the clinical potential of understanding these precise, timed gene programs?
The clinical hope is enormous, particularly when you think about devastating memory-related diseases like Alzheimer's or other forms of dementia. Currently, many neurodegenerative diseases attack the initial memory formation center, specifically the hippocampus, relatively early in the disease process.
Which means traditional therapeutic research is often focused on trying to stop the initial damage where the memory starts.
Exactly. But if we understand the sequence of molecular timers, it suggests a completely different and really powerful therapeutic strategy: bypassing damage.
Bypassing damage. Yeah. If scientists can identify the second and third areas that are important for memory consolidation, the thalamus and the cortex, and understand the exact molecular commands they require, like Camta1, Tcf4, Ash1L, they may be able to redirect memory pathways around damaged brain regions.
So if the hippocampus is slowly dying and failing to form new memories, we could potentially use targeted drugs or gene therapies to stimulate TCF4 or Ash1L in the healthy parts of the cortex directly.
Forcing the stabilization program to engage and take over the consolidation function, that is the immense potential.
That's incredible.
Dr. Rajasathupathy suggests that if we know the second and third areas that are important for memory consolidation, and we have neurons dying in the first area, perhaps we can bypass the damaged region and let healthy parts of the brain take over.
It offers a powerful new avenue for intervention by focusing on the active stabilization phase, the clockwork, rather than just desperately trying to save the failing formation center. It's like a molecular backup plan.
It is. This could fundamentally change drug targeting. Instead of focusing on neurotransmitters or plaques, we could be looking at molecules like Ash1L as key targets to reinforce existing memories or help establish new ones, giving patients a second line of defense against neurodegeneration.
And when you think about the implications for treatment, if TCF4 is crucial for strengthening the physical connection,
And we know that connection is vulnerable in Alzheimer's.
then targeting TCF4 might make those connections physically more robust, prolonging memory functionality even as the initial input source deteriorates.
It's an approach that targets resilience rather than just a cure.
Okay, so finally, what are the next crucial steps for the research team? The picture is incredibly detailed, but where are the remaining gaps in our understanding of this memory timer system?
Their ongoing goals focus heavily on uncovering the trigger mechanisms. We know these molecular programs turn on sequentially, Camta1, Tcf4, Ash1L, but they need to figure out exactly how these molecular timers are activated.
What's the specific electrical or chemical cue that initiates Camta1?
Exactly. And what signal precisely must be sustained to transition from Camta1 to TCF4 and ultimately to Ash1LA? They're trying to understand the nature of the molecular starting gun.
And most philosophically, I suppose, they need to fully characterize the importance metric.
That's right. We know that in the mouse experiment, repetition was the stand-in for importance. But in a complex human brain, importance is multifaceted. It involves intense emotion, threat assessment, survival relevance, novelty.
All these other factors.
Right. They need to determine what mechanisms the brain uses to truly evaluate the importance of a memory and decide its necessary duration. Is there a single signal that flags a memory as Ash1L worthy?
And their ongoing work continues to confirm the thalamus as the central hub for this decision-making and its parallel, sophisticated communication streams with the cortex. They are clearly looking at the life of a memory beyond its initial formation in the hippocampus, viewing stability as a marathon, not a sprint.
It's a complete shift in perspective, viewing the brain as an extremely sophisticated timed molecular reactor rather than a simple electronic circuit board.
So what does this all mean for you, the learner, and for how you view your own history? We started by questioning how a passing impression becomes a defining memory, and we've drilled down to the level of precise genetic regulators.
I think the key takeaway is that dramatic transition from the simple on-off switch model to this highly coordinated, actively time-dependent system. Memory persistence isn't an instantaneous binary choice. It's a demanding construction project managed by a precise sequence of molecular timers.
Camta1 and Tcf4 in the gatekeeping thalamus, reinforcing the structural connections.
Culminating in Ash1L in the cortex, promoting that chromatin remodeling for ultimate decades-long stability.
And this isn't just cutting-edge, obscure neuroscience. It fundamentally changes how we view learning and retention. If forgetting is the brain's default state, then memory is an active process of time stabilization that has to be earned through sustained repetition or extreme perceived importance. It confirms that the way we interact with information, how often we review it, and how actively we engage with it, is directly manipulating our own molecular destiny.
And that leads us to a final provocative thought for you to consider. The original experiment used repetition, the frequency of exposure, as the brain's internal signal of importance that triggered these molecular programs.
Right.
So if repetition is the trigger for the molecular timers that determine memory longevity, consider how human attention, conscious effort, and space review might actively hijack or influence the activation of these very same Camta1, Tcf4, and Ash1L molecular timers.
It raises an important question then.
It does. Can we consciously choose to engage the full stabilizing mechanisms that determine the longevity of our personal history? By consciously focusing, reviewing, and applying effort to information over distributed time, we are likely manually influencing our molecular clockwork, moving our knowledge from the temporary Camta1 system to the permanent, architecturally sealed Ash1L program.